These are a nice opportunity to get together to learn about what our colleagues are doing. This is how we keep track of what's going on, and if we have common interests and that sort of thing. We have 6 speakers today from the college. It's also an opportunity for us to celebrate people's success. So here are our speakers. They're going to talk in this order. Vivian Powell, this year was named the Writer in Residence for the Buckhannon Fine Arts Project. Alexandra Kuzmanov recently was awarded a NIH grant, and she's going to tell us about that. And the rest of these are related to NSF funding. So a lot of great accomplishments. And so we really need to celebrate this success. So I think, why don't we celebrate this success? Everybody's going to do about a 10-minute talk with about 5 minutes left for questions after that, okay? So we'll go through Matthew.
All right, I'll be the trendsetter here, try to keep us on schedule.
I have the...
Oh, okay. Corey will keep me honest here. So I'm going to quickly go through. I'm going to try to fly through here. Biography, just for people who don't know who I am or want a refresher on where my background is. I work in the area of mathematical biology, emphasis on the mathematical biology. I'm from the math side there. So I do work in 2 fields, biochemistry and epidemiology, both from the math side. So I'll talk a little bit about work that I've done recently in both of those areas and ending with how I'd like to bring those 2 fields of work together. Because I think there's a lot of opportunity there. So quickly, just who I am. There's another picture of me. So my mathematical journey or my academic journey, so I'm from Canada. I did my degrees there, Bachelor of Arts in Mathematics, Master's and PhD in Canada. Moved to the US, I did a postdoc in Wisconsin. Then I was tenure track out in California at San Jose State University before getting the opportunity out here and coming back a little bit closer to home. But I teach sort of the applied math curriculum here, which is what we have. I do research, and as you'll see. So that's sort of where I'm coming from my journey here. My one field that I work in is mathematical biochemistry. The like elevator pitch on that, which is about what I have time for, right, is if you look on the right there, you have a picture of a very, very crude overview picture of what a cell does. But what a cell does, you can kind of think of it as like an information processing unit. I mean, what it does fundamentally is it takes inputs, right? It processes them, and then it has to do an output. Right? Whatever a cell function is, whether it's signaling another cell to do something like a neuron or something like that, or whether it's, if it's a single cell organism like a bacteria, it needs to activate the flagella to chase after something it's chasing, whatever. And inputs and outputs, it's kind of like a computer, right? But if you think about how a cell does that, it does it with chemical processing, right? It has a bunch of chemicals that are in there that will transduce these signals from the input signals to the output, try to get the appropriate protein configuration in there to get the response. So it's a very interesting system where you've got a computer that doesn't have hardwired input, output, like through wires and signals, binaries, and it's not invented those sort of things. It did it with basically just a chemical soup. And it somehow faithfully produces input-output signals, or somewhat faithfully. I guess we shouldn't say always faithfully. There's a lot of work in this area. I've had about, I guess I put it down there, 14 years of experience since grad school working on problems mathematically and how you build mathematical models of these. I've got about 24-ish publications in that area. I also work in the area of mathematical epidemiology. That's been basically since 2020. Something happened in 2020 that kind of piqued my interest on the epidemiology side. So I have since then, I've done a pretty hard pivot to mathematical epidemiology, where basically the idea is you want to predict what happens next and understand what has happened so far and why in order to understand and predict what's going to happen next, potentially under a variety of circumstances. My pitch here is we get to, right, so if you think about like your possibilities, right? Lived experience, we get to live once. If you want to do like a controlled like scientific experiment in a laboratory or whatever, you get to run it a couple times, but it's kind of expensive. It takes a lot of time and resources and that to like, I'm gonna run the same experiment 20 times and then see which one's better of those. But then you get to select, so I'm gonna pick the best of those 20 runs, and that's the reality I choose to live in if I've done things correctly. Mathematically, if you've got a computer and a good mathematical model, you can run millions of different simulations on all your hypothetical future scenarios, and then you get to pick your best one. And it's cheap. Computer resources are cheap, and also mathematicians are cheap, as it turns out. So we do a lot of stuff in this area. I'll tell you very briefly about some of the things I do. I do have a current NSF grant that'll be wrapping up this August. So either maybe get another year if there's enough funding left over and NSF says okay. I'm currently funding 3 students. I do have a few students who sort of wrapped their stuff up that I've also funded. But that's been since 2022. I'm going to try to keep track of time here. I'm doing okay. But anyways, I wanted to get into like what I actually look at like a basic level so you can understand what I work on. In either of these spheres, so this is a biochemical example on the left. On the right is an epidemiological example. All right, so on the left here, I've got basically, imagine some biochemical things like in your cell or whatever. I'm imagining a very simple system here where the E and the F are basically their enzymes, they're proteins that will modify other proteins, right, which is what the cell always does. So it takes in a substrate, binds to it, produces some product, so it modifies it in some way, which is how signal transduction happens. Those signals are carried in the cell. And then you have another enzyme, a kinase, or phosphatase, depending which way you're going, that will take that modified thing and regulate it, turn it back into its original state. Seems like it is kind of defeating its own purpose to do this, to turn S's into P's and P's into S's. But this is how metabolism is maintained in the cell, right? They do all these active shifting around of things to keep things at a stable level of the concentrations you want in there. You model this mathematically, you get a system, at least in one modeling thing, is differential equations. If you've never heard of differential equations, don't care about it, don't worry. Basically, it's applied calculus. These are all derivatives over there, so if you can understand rate of change, you want to figure out how these things are going to change over time. That tells us what's going to happen next, right? That's what we're always trying to do here. Right, so again, even a relatively simple system here that underlies a lot of biochemical processes, fairly complicated-ish. I wouldn't throw this to an undergrad class, for instance. But it's stuff you can handle. And you can write it down, it's kind of cool. You have a similar sort of thing with infectious disease. If you want to model that and say, how does an infection spread in a system? You divide your population up into people who don't have the disease, people who have the disease, and people who have recently had the disease but have some immunity against it. And you say, how do they interact? Well, they interact in some way where there's going to be some transfers. There's going to be an infection, right? An infectious person could infect a susceptible person, make them infectious. And an infected person could recover because they spent enough time quarantining, their immune system took over, and now they no longer have the disease. So you can have, even with just transmission of the disease and recovery, we get a system like this. Right? So those are the kind of models. They look kind of similar-ish, both differential equations. Where I've done my recent work in epidemiology has been on things like this. So this is the spread of variants since the start of COVID. Right? So you'll notice here, this is about, Omicron's right about there. So that's where the last time we kind of heard about it en masse, like, oh my God, everybody panic, it's changed, it's the whole new thing, and it is. But there's been a lot of transitions since then. Right, and we're trying to understand why that. I've been looking specifically at how vaccination coverage levels can influence actually the strain over strain competition, right? So not necessarily claiming any one of these is directly a result of that, but is it possible, right, that you introduce a vaccine and depending on how the different competitive balances between those different strains that are in the environment, it's, the vaccine's a little bit more effective against this one versus that one, or maybe it's the same. Can that make a difference in which strain wins out in this picture, right? It turns out you can, but you got to build a model first. So built a relatively simple, not overwhelmingly complex model. But you'll notice I break things apart into 2 different strains. I also have different dynamics here so that the different strains can have different interactions. These are the vaccination edges there. Don't have time to go into too much of the details, but you can do the same thing you did before, where you build a differential equation model out of it. You can add realistic delays if you want. So if you want to say it's like, oh, hey, when somebody has the disease, their immune system is going to, that immunity is going to wane over time. Well, how does it wane? You can pick the distribution that you want it to wane by. You can go to the experimentalist and say, it's like, well, How long do people have immunity from this after they get it, after they get vaccinated, whatever? You can choose different distributions. And so these are the terms there. But no need to worry about the details. You can analyze it. And you can see that actually the vaccine level in different circumstances can have a very big impact on which strain wins out. So this is the two strains. Here you can see is the vaccination level. That's vaccination level going up this way in the population. And you see one strain sort of taking over from the other. So we could analyze two different cases. I'm not going to have time to go into much of the details about why these two cases are different. But you can get the switching to happen in either order. Right, we're one strain, so I've got one strain set to be the more transmissible strain, one strain set to be the more immune resistant versus transmissible strain. And you can see there's a difference in which one will take over. But the take home is, it's complicated, right? Just because you can't just sort of say, it's like, oh, I'm going to do this one thing, and the consequence of that thing in this relatively simple system, I know it's going to be this one thing. Everything's going to die out or whatever. Even when things are working the way they're supposed to, and a vaccine in this case, in both cases here, is effective, you can see a switch and sort of what you'll see at the population level. Like in this case, you'll see one strain take over from another, not because the vaccine isn't effective. In both of these cases, the vaccine is effective against both strains, but because it's complicated. Right? So you need to pay a lot of attention to the details of what you're doing. And you can't be surprised when you see something happening and going, I got to throw it all out. Right? It doesn't work. Right? It's complicated. So another thing on the biochem side that I've been sort of working on, inspired by my recent work in mathematical epidemiology, you've had this number that you've probably heard about, probably haven't heard about it as much recently, but have certainly if you dig back a few years, the basic reproduction number, which is basically a measure of how quickly a disease is going to spread, the number of new infections per existing infection. Well, can I use that in biochemistry? Right? So these are very similar. You look at the models from several slides ago. The models are almost the same. They're just differential equations, things multiplied together. There's not very much difference here. And there's a big open problem in biochemistry, which is how do I get stability of species? How do I get things to guarantee that they're going to either exist in the system or not? So you might think about a metabolic system where you have a finite number of metabolites. Are you going to exhaust those metabolites? That is analogous to an infection dying off. It's something mathematically, it's just something going to zero. Can something go to zero? Well, surely the mechanism here, the R0 value, tells us in mathematical epidemiology whether something will go to zero or not. Surely there's some analog in biochemistry. Not been very studied, but I'm looking into it now, and I've got sort of a draft of a paper up. It works. You can do this, but the theory's got to be cleaned up. But you can basically take that basic reproduction number, transfer it over to biochemistry, and hopefully answer some meaningful questions that other people are interested there as well. But got more work to do there. But you can do things that are a lot more complicated than you would expect. So this, if you had to do this directly to figure out whether you have stability, things can go to zero or not, you'd have to take at least most of these variables, at least a 7x7 Jacobian, and then find the determinant of it. It would be practical. You wouldn't be able to write it in 100 more slides, basically. The computation you run on your computer, but like, I don't want to store that. You can very quickly do this by basically modifying what they already know to be true in epidemiology, borrowing their methods, adapting them, and using them here. And you get, this looks bad, but believe me, it's better than the alternative. So I'm not going to talk much about this because I am, I think, really out of time. But I would like to, this idea of building bridges between epidemiology and biochemistry, adapting, seeing where techniques from one area can be adapted to the other in just novel ways, seeing what can be cleaned up. That's sort of my idea drafting for my next NSF grant proposal, which I hope to get out in the fall. But yeah, I think that's it. Any questions? I think I'm time for maybe a minute of questions or something.
How predictive are they? Like, are we using mathematical models to actually say, like, yeah, this is the one that's going to predict what's going to happen, or is it sort of like after the fact and say, this is the model that best explains what happened?
What a great question. What a hard question. So if I had more time, I'd have another slide where it's like, and here's all the reasons why you shouldn't trust this, like without a little critical thought. And there's nothing wrong with the mathematical models. Mathematical models are really good. In a lot of cases, they can be really detailed and get a lot of the factors that you want in there. But there's incredible sensitivity to what's actually happening in the field. That by the time you change, you say it's going to happen this way, and by the time you say that, something has changed out there, right? And there's such sensitivity to the parameters, the initial conditions, right? You need to basically have great data. Great data. We do not have great data. We don't have data that tells us on this day we have backlogs of months worth of data that are aggregated in certain ways where you don't know when something happened, right? So it's really hard to in practice use that, but there are lots of people. There's so many people. I was at a conference virtually 2 weeks ago where the whole question was like, how do we get better predictive models out there faster so that we can make our actual on-the-ground responses less generative of the skepticism that was generally the general feature during the COVID response, because it was like, you said it was going to happen this way, and then something changed, and it didn't happen this way. It's like, nobody's up for that. It's like, yeah, but we also said this stuff by the time you got there that far. It's like you've lost them, and so it's hard. It's a challenge.
Thank you.
Great. All right, hang on there. For the recording, I guess.
It's for the recording.
Yeah.
Yeah, we do need it. There's a lot of extra noise. That you can just clip anywhere.
Okay.
Yeah.
Okay. Good.
All right.
Okay. Can I put this in?
Slides right there.
Thank you.
Oh.
Can I just? Is that USB? Oh, should work. Oh, awesome. Okay. Thank you. Okay. Hello, everyone. I'm Alexandra Kuzmanov, Assistant Professor of Biology. Here is a little bit of my education there. I have a broad background in biomedical background, and I've been always interested in reproductive toxicology, reproductive health in general. My current focus is effect of environment on reproductive health or fertility. So I bet you know at least someone struggling to get pregnant, and in fact there is 19% of women not being able to get pregnant after 1 year of trying. So for several decades now, we've been learning about the effect of environment on reproductive health, and became increasingly clear that chemicals in the environment can affect our health. You can get exposed to chemicals from environment in different ways. It could be through skin contact, through ingestion, even inhalation. My current focus is plastics. This is one of the environmental contaminants. I don't know how much you know about plastics, but plastics is made out of polymers, and there are additional chemicals, plastics additives, that actually enhance performance of plastics. We're all exposed to plastics every day. There are some plastic bottles, even that can seems to be metal, but cans have plastics lining inside, right? So not only that, plastics degrades over time, and in the environment you now have micro and even nanoplastic particles that we can even inhale. So we're constantly exposed to tons of plastics. One of those additives that I mentioned enhance performance of plastics is actually bisphenol A, or BPA. And that's my current focus. Over years, we learned that BPA, we've been using BPA since 1950s, and over several decades, we actually learned that BPA can affect, indeed, is toxic to us and affect our not only reproductive health, but among other things, reproductive health as well. BPA could interfere with hormones, so hormone signaling, but when it comes to reproductive health, actually could decrease quality of reproductive cells, eggs and sperm. So we learned this several decades ago, and then manufacturers started replacing BPA in plastics. So you find these BPA-free bottles, basically no BPA, but they needed something else to support performance of plastics. They did not go far away from BPA. Currently, what you have instead of BPA in that plastics are BPF, bisphenol F, and bisphenol S. And if you look at the structure, it's just a tiny change, right? So there is these methyl groups are missing in here. And then there's trying to think, I'm not a chemist, just biologist. So a slightly change, and there is accumulating evidence showing now that even this bisphenol F and bisphenol S affect our health. So there is a high need for structural innovation of bisphenols, and this is where Dr. Timmons and I teamed up on this project. Dr. Timmons is a synthetic chemist, I'm a biologist, so what we're doing, we're trying to find a safer bisphenol. So she's synthesizing novel bisphenols, changing a little bit structure of BPA, and I'm testing in my lab if they're any better than BPA. So, how I test this. This is my model organism zoomed in. This is actually a tiny worm, nematode called C. elegans, Anaerobditis elegans. We maintain them in these plates. So this is an adult, which is 1 millimeter long, tiny worm. If you zoom in here, you see something like this. We use these worms to test chemicals. We'll probably ask how you can test something on this worm and then apply it to humans. So why we like to use these worms? Well, they're cheap, easy to maintain in the lab. Their life cycle is very short. They go from an egg to an adult in 2-3 days. They live only for 3 weeks. If you want to test a chemical and see if it affects development or their lifespan, quick result, right? In a week, couple of weeks, you get your result. So, but how we can apply this to humans? You can't really test everything, but when it comes to toxicity of reproductive tract and quality of reproductive health and quality of reproductive cells, you can definitely use it because this process is conserved from yeast, the baking yeast we all buy and use. Meiosis, process how we make reproductive cells, is conserved from yeast to humans and worms as well. So you could test how chemicals affect eggs and sperm. They do have 2 sexes, males and hermaphrodites. Hermaphrodites do both, oogenesis and spermatogenesis. They actually switch. They do first spermatogenesis for a short period of time, and then for 5 days they do oogenesis. Males do spermatogenesis all the time, so that's how we test chemicals using these worms. Okay, so this is how we test this, and when we started this collaboration, first what we did, we tested these 2 bisphenols. You see the change. Instead of small changes in here, Dr. Timmons put these bulky groups on the side. We were hoping that this is going to help, right? It's a huge structural change to me as a non-chemist. However, when we tested this, these were equally toxic. So BPY and BPZ, I won't go much into details in here. This is control. These are different bisphenols. Here, control bisphenols. But basically, what these results showed that the number of progeny was decreased following exposure to these bisphenols, showing that something was going on with meiosis. Obviously, that's why number of progeny. These worms, I didn't say that. Each worm has 300 progeny. Each hermaphrodite. And then also the quality of reproductive cells were lower because some of these cells, what happens during meiosis, if the cells are affected by a chemical, something's wrong with them, they undergo programmed cell death. So this is the number of cells undergoing programmed cell death, showing, okay, some of the cells were not good. So it was comparable to commonly used BPA and BPS. So we wanted to switch gears, and at the time when we wanted to decide what to do next, there was a paper published that this chemical seemed to be a bit better, tetramethyl bisphenol F. So this is not commercially available chemical. We read the paper. They said that the chemical shows lower interaction with hormones. It was just this was industry related. We were a little bit skeptic, but we're like, okay, let's take it. Dr. Timmons made TMBPF, and we decided not only to make this one and test, we also decided to make more. Only 3 minutes, okay. So this was the proposal. This is our current project. She proposed different analog similar to this TMBPF chemical. I proposed tests, and we got funded. This is our current project. In the meantime, we tested TMBPF effect on male fertility. We published this paper recently. TMBPF, interesting thing, it is a little bit better. We showed that it did not affect much male fertility in C. elegans. I won't go into details, I don't have time. But what we did show eventually is it affects embryonic lethality, which means if you see that some of the embryos did not survive, there are some chromosomal abnormalities. So yes, it's a bit better, but it's not perfect. So that's what we're working currently on. Testing these bisphenols. A couple of days ago, actually, we got additional funding. This is NIH funding as well, but it's our collaboration with Wayne State. CURES supports community-relevant projects. It gives funding for 1.5 years for you to get preliminary data so you could apply for additional funding. And we got this, what we're doing, we're actually exposing worms to those bisphenols and looking at all the proteins. This is a comprehensive analysis of all the proteins inside them, basically comparing protein profiles, looking for genes that are affected by bisphenols. Why? Bisphenols are, and this is, I'm going to finish with this, bisphenols are quickly metabolized. We excrete them to urine, but it seems some research recently showed that they do have long-lasting effects, changing gene expression. And we want to know which genes are affected and how that can affect later our health. So I'm going to finish with this. Just thank to my previous and current students who are working on this project, and you for listening, and I'm happy to answer any questions. I was very clear. Oh, it's not gone.
I got a question.
Oh, sorry. Go ahead.
So you say a lot of your research is in reproductive health.
Mm-hmm.
Do you have problems getting funded with that because it's women's reproductive? Do you know what I mean? Because it's
So, yeah, it's hard to say because I just, started, when I joined LTU, I switched gears. I was working on other things. I was actually working on spermatogenesis when I worked as a postdoc. Before that, on a stress pathway, my first time. So,
okay.
we'll see.
It's not related. So we talk a lot about doing like
Feel
our research
intelligent.
in our classes.
Uh-huh.
What classes would you talk about this in?
I teach biology,
So like,
but
is there a specific biology class though where like this is?
we actually do some of these essays in cell biology laboratory and cell biology lab. We discuss this, but yeah. This is supposed to be a video, but it's not working. Video is not working, but that's fine. Should I leave it for other people, or do you need this, Vivian?
I only have 3 slides.
Oh, that's okay. Thank you.
That microphone.
Oh, yes.
Thank you. You can just clip that anywhere you want. I'm just going to put this right on your sweater.
Sure.
Can I just put it in my pocket, or does it
Yeah, you can put it right in your pocket.
does that work? That's not gonna.
That works, yeah.
Okay, cool. All right. Cool. All right. Hey, everybody. My name is Vivian Cow. I'm Associate Chair of the Humanities Department. So this year I have the privilege of being the writer in residence at the Buckham Fine Arts Project. So the Buckham Fine Arts Project is an artist collaborative based in Flint, Michigan. Even though there are artists from other places who also participate in the collaborative. But it's a nonprofit, and they run, one of their main things that they do is that they run this gallery. So this is the Buckham Arts Gallery in Flint, Michigan. It is open Wednesday through Saturday if you want to go and check it out. It's very cool. So this is the gallery space. And what the writer in residence does is they write a literary creative piece It can be fiction, it can be poetry, it can be experimental, for each of the exhibitions that comes through. So they usually have, they switch out their exhibitions every month, and every month they will have 2 or 3 concurrently running exhibitions. It's not a big space, but they're like really amazing at being able to use the space to accommodate 2 or 3 different exhibitions. Sometimes they're group shows, sometimes it's one artist. Some of the shows are national juried exhibitions. It can be any number of things. So I thought for my few minutes with you, I would give you a sense of actually the art at work, or what it is that I do. So this piece is a piece that came through November through December. This was part of a national juried exhibition called Brown Bag. So a whole bunch of artists made art surrounding the theme or responding to the theme of brown bag. And so this was one of the pieces that was chosen to be exhibited. And I do have to ask that you guys don't take photos of the art or reproduce it, because I did not get permission from Mabel, the artist, to show this, but I did want to show you like kind of what the process is. So I do have to ask no circulation of the visual art. So this came through November through December 2024. It just left. And what I do is when they put the show up, I go in with my little pad and pen, and I look around to see what kind of inspiration I get. And my job is to do ekphrastic writing, which is writing that responds to the art. So when I saw this, I was immediately drawn to it because it is a print that shows the process of making dumplings. And making dumplings is something that is a really special thing for my family. And when I was growing up, I always remember like holidays, birthdays, whatever excuse we could find. My dad and his 6 siblings, my dad had a very large family, would get together and everybody would make dumplings. And it was like a big assembly line process. And I wanted to write a piece that kind of expressed that kind of family love and the kind of industrial nature of our dumpling production. So, I mean, I'm talking hundreds of dumplings would come out of this assembly, like 400 or 500 dumplings, right? Okay, so this is the print, and then this is the piece that I wrote for Folding Gold. So I titled mine Folding Gold as well. For the pieces that I write for the shows, I often do just use the same titles to signify that I'm responding to a particular piece of art. So, all right. So this is the poem that I wrote in response to that piece. Folding Gold. Starts the night before. Ground meat of all kinds, set in plastic tubs, cabbage, garlic chives. Glass noodles, tofu, egg, shiitake salt. No food processor, just sibling labor. Father mixes dough, sets to rise till morn. Uncle rolls it out, cuts even morsels. Back forth, rolling side to side. Toss disc to first aunt, second aunt, third aunt, fifth aunt, sixth aunt, and youngest aunt. Never run out of sisters, but run out of dough. Brother, where art dough? More coming, brother dear, always no patience. You always slowpoke since you were little. All good for nothings. This last from Mater, old but still top chef. Supervise with frown, eyes not miss a thing. Not a drop of mix wasted, not one extra wrap, ratios ancient. How could they be wrong? If wrong, your fault, said to her useless brood. Old too, but still bad at making dumplings. See? Ugly, too big, too small, too fat, will break in pot. Just wait. No, Ma, they won't. Not this time. Go sit down. We got this. You got nothing but broken dumplings. You will see. 14 hands, knead, roll, cut, toss, fill, pinch, pull, shape, set. Folding gold on old gold mountain. In home not home, home away from home. Home, if Ma here and we here. Dumpling pasts and dumpling futures, all laid out on table. Covered in flour, smelling of garlic, steam rising from pot, too many voices. In the beginning, Old Man Heaven said, "Let there be dumplings, and eat them while hot." So a little bit about the form of the poem. The form is modeled after a classical Chinese poetic form, which consists of 4 lines with 5 syllables in each line. So in the classical Chinese form, the entire poem would be like one of these stanzas or little paragraphs. So you get 20 syllables total, split up between the 4 lines. So what I did was I just kind of adapted that to the stanza form, and I try to. There's some lines in there where I kind of cheat a little bit because it's just it's hard in English, but I tried to keep it to that form to kind of be I don't know, just sort of to call out a kind of inheritance that is passed down from, in immigrant families, even across languages, right? So in Chinese, this works a lot better because you can convey a lot with a single syllable. So all the characters are a single syllable. Many words are also just one syllable. So you can like say a lot in 20 syllables. English, it's a little bit harder. So eventually, at the end of the year, so my time with the gallery will run September 2024 to September 2025. And at the very end of the year, all the poems and all of the artwork, so photos of all the artwork, will be collected and published as a book. And hopefully that will be out in like February, March 2026. So that's it. Yes, Franco.
Yes, I like very much this interaction between forms of art. It's wonderful. My question is about the process.
Yeah.
So, I suppose there are many pieces from different ages, right?
Yes. Yeah.
Okay.
Yeah.
Is your task or your work related to all the pieces, or you select? How many do you select? And
Yeah.
how much do you work in every piece? How many works you are going to have at the end of the experience? If you have any kind of prediction.
Yeah, no, that's a good question. So the requirement is just at least one poem or one story or one creative piece per exhibit. So if like when this show came through, there were 2 exhibits. One was called Brown Bag, and the other one was called like Diners, Dives, and Drive-Ins. And so my job was to do like one for each. But sometimes, I'll be inspired by, 3 or 4 pieces, yeah. Or, I'll write a bunch of shorter pieces. So it's really up to me. But the requirement is at least one per exhibition, yeah. If it's a group show, like both of those were group shows, I try to do more than one, just so I'm engaging with, more than one piece. If it's a solo show, sometimes I'll just do one, because it's only one artist. So.
Every time do they change the exhibition? I didn't get that. So the brown bag, how long it lasts?
1 month.
1 month.
Yeah,
Okay.
yeah, or 4 weeks. Yeah, so
It could be
they.
around 12 different exhibitions.
Yeah, I think they do 11 because they break for it must be for the Christmas holiday. But yeah, so it's really exciting. Like every month there's something new, and then I go in and it's like the gallery looks completely different. And it's all volunteer run, and so I work for free, and then all of the proceeds of the book sales at the end go to the gallery. So it's kind of like public intellectual public service, I guess, if that makes sense, or a creative, creative scholarship in the service of the community, because all of the dollars generated will go to running the gallery. So, yeah. Yes.
How do you interact with the visual artists who, from the other side of the creative people? How do you interact with them?
Yeah, so unless they're local artists, usually I don't meet them. Yeah, because usually what they're doing is they're just sending in their work, and then the gallery, the collective, I guess, chooses the pieces that they want to display. And they might have some, back and forth with the artist, but I don't, really communicate with them. So they don't see the work of the writer in residence until the very end. Yeah. But if it's like, if they're like local artists and they're like my friends, then, I'll like show them my pieces and we can talk about it and stuff. But yeah. Cool. Thanks. Yeah. All right. My pocket.
Okay. Oh, thank you. I don't know what happened. They got so big, the file size. I'm sorry about that. Yeah, I noticed that when I sent it. I was like, oh no. Okay. Alrighty. There we go. Okay, so yes, my name is Bruce Pell. I'm an assistant professor here in the mathematics department. Today, is there a clicker? Or should I just?
I'm sorry.
Oh, that's fine. Oh. Oh. Oh, okay, thank you. Just 1 second here. Here we go. Okay. So like what I was saying, I'm a assistant professor in the mathematics department here at LTU. Today I'm going to be discussing real quick some of the research projects that I've been kind of working on the past couple of years. Some of them are similar to our first speaker, Dr. Johnston, Matthew Johnston. Me and him actually, we collaborate on a lot of things. So I'm also like a mathematical biologist. I work mainly more in epidemiology, and so I'll talk about some of those projects. And then the last section I'll talk about more about like the more current and future work that I've been kind of working on. But before all that, I want to also just point out, right, the projects in this talk, they're from a whole team of, like an interdisciplinary team of researchers across the US. So we have people from Arizona State, right, in mathematics, Oklahoma State, geography, in biology, down the street at Oakland University, biomedical engineering and mathematics. So there's a lot of people coming together to try to answer some of these questions in these projects. Myself, I'm from Michigan. I was born in St. Clair Shores. I went to school for my undergraduate degree at Oakland University, and then I went to Arizona State for my PhD. And like I was saying, a lot of my research is in the interface of math and biology. Okay. So what do I actually work on?
Currently been working on wastewater-based models of infectious disease. So Matthew was talking about earlier about how the data is so bad, right, in trying to understand the spread of a disease. What new types of data should we explore? This more recent type of data is wastewater-based. And these projects are funded by the National Science Foundation. The first one is based on just incorporating the data into the math models. And then there's a second grant that is taking those models and then incorporating human behavior. And so like social distancing and frustration and those types of mechanics. The second project, I am looking for students. So I just wanted to point that out. And later in the talk, I'll talk about Taylor Hushke, her project. So why wastewater? So it turns out that wastewater, it's anonymous. That's the first thing, right? The data collected is anonymous. And what type of data I should be talking about? Like, are we collecting from wastewater, right? So when you get infected, you excrete, you shed the virus particles for certain diseases into the wastewater system. And we can gather that and sample that and then get an idea of what diseases are in the population that sewage serves. And the nice thing is that, yeah, it's anonymous. There's no lag either, at least perhaps a reduced lag, right? Instead of reporting cases that are gathered from hospitalizations or things like that, right? Which takes time. And so that's kind of the motivation. And it's more data to try to calibrate the models so that the models are hopefully more accurate, okay? Yeah, so I just wanted to put this slide in because they actually saw that there was polio in 2022 in New York City, and it was detected by wastewater techniques. And wastewater's been around for a while. It hasn't really it's become more popular since COVID, but before COVID, it was used for monitoring specific types of drugs that were in the population and things like that. So it's a way to survey and some type of surveillance system for diseases. Here's how it works. I kind of already explained it, right? There's an infectious person. They infect a bunch of people. Those individuals, they go and they shed virus into the wastewater system. We collect and sample the wastewater at some treatment plant. And when we have that data, we can then use it in our biological experiments, our mathematical models. And so that's kind of what I'll be talking about today, okay? All right, so yeah, how do we leverage this new data set? So here's a mathematical model, SEIV, right? Kind of like what Matthew was talking about earlier, right? S is susceptibles, E is exposed individuals, I is the infectious people, V is the total virus that's in the wastewater, okay? And for COVID, we know some of these parameters. We know right, the latent period is 3 days. How long you're infectious is approximately 8 days. We don't, however, know like the transmission rate, right? The probability of you going out, meeting someone, and then the probability of you making contact that's infectious, things like that, okay? What we do know, though, is from hospital data, we know the shedding curve of an average individual. And so this is data that's been collected in 2020. There's more updated data sets now. But what it shows is, so at day 0, that's when you first get infected. And then as the infection progresses, right, the amount of virus in your body shoots up to a maximum, and then it slowly goes down. But that's over a long period of time, okay? And so the infectious period is what's shaded there. It's like 3 to 11 days, right, post-infection. And so we can use that to figure out the average number of virus particles that is shed by a single person, okay? So we know how much is shed into the virus or into the wastewater system, and then we can use that to inform our mathematical model. So here's a simple model. This is the function for that curve that you're seeing there. And then we take this information along with the data sets from in this case, it's from Massachusetts. And so this is one example of the treatment centers. And this is Fu Xing. He's the biomedical engineer who works with the people at the wastewater centers, and then talks with the modelers. So we fit the model to the data, right? We only fit the virus equation to the virus data, right, in the wastewater. And then we use the math model to project what is like the total number of cases, or the number of cases per day that are actually putting that much virus into the wastewater, right? So it's a way to kind of analyzing and projecting how many actual cases there are. And what we found was at least 10 times higher than what's actually reported by the CDC. And so what you see here is, in the red, there's the CDC data on the right panel, and then the model projection of what is being predicted by the model. So it gets us around of like not having to worry about, the people that don't like self-select and tell the CDC, right? That they're infectious, right? I know like there was an app that you could have used and you could report yourself to the CDC. Not everyone does that, right? But the wastewater modeling, it gets rid of all that. We don't really need that information because the data is in the wastewater, okay? There is some more things to think about here, right? And this is just a very simple model, right? In the wastewater system, there's the temperature changes, right? And temperature will affect the degradation of the virus particles and things like that. So there's a lot of information there, right? A lot of science that needs to be done. We went a little further and we incorporated temperature effects, and that did actually reduce the prediction to like 8 times higher. Okay, so there's definitely room to kind of to improve, and I don't have the slide here, but how do we validate this? And well, there's other groups around the world that are working with wastewater, and we show our comparison. Well, we take our model and we compare it with theirs, and that's one way to kind of start validating the whole modeling community. So that's with one strain. Matthew was talking earlier about, well, there's multiple strains of COVID that were going through the population, and so we were interested in taking what we had and using a 2-strain model. So you have 2 I's, I1 and I2, right? Different strains of COVID. And I'll skip that slide. But we were able to do the model fitting as well, and it gave us a nice prediction of when one strain was overtaking another, and you could see it in the actual sewage data, in the virus data. And then you can see the actual the projected, in the middle panel, the projected numbers of daily cases, right? And you might notice, why does it kind of get closer to the CDC data at the end? That has to do with people that recover, you're still actually shedding virus. And so there's a lot of like interesting things going on there. Even though you're not infectious and you're not infecting people, it's been shown that people like 20, 30 days past infection, they still shed virus into the wastewater. So there's a lot of interesting math, a lot of interesting science that needs to be done. My current and future work, just real quick, this viral curve that I was showing, we want to model that instead of just using a simple function. We want to model that with a whole system, right? A mechanistic system that describes the gut and the lung and the dynamics of the virus infection. And so Taylor Hushke, she's been working on this with me, and just recently she got really a nice model fit to some of the viral shedding data. And now we're really interested in seeing how do you control that shedding data, right? That curve. Because now we have the mechanistic model with all the biological parameters, we can see what changes what. The last thing is nothing to do with COVID, but the last thing is temperature changes affect infection progress. And so my other project with Thomas Raffel down at Oakland University, we're interested in seeing how does temperature effects change host-parasite interactions. And so there's a lot of interesting things there. He's a biologist. He's infecting frogs, and then we're running some model simulations. And that is it. Okay. Okay. Hopefully. Any questions? No? Franco, yeah?
Yeah, I think this is really fascinating. I would like to, I don't understand how you can separate, for example, a person that is a new you just have a number, right? From the sample that you have a number.
Right.
And you have a number the next day, and then a number the next day.
Mm-hmm.
But there are new people that are getting infected.
Right.
So, and other people that still have a certain 70%, 60%, fading.
Yeah,
but
yep.
other new are coming. So there is a system of prediction that you are using to separate these two sources.
Yes,
of
yes.
The number of viruses.
Right.
That you see in the world.
That's the power of the math model, because the math model describes that process. Yeah.
Because if you have more, it must be that there is new infections.
Oh, I see what you mean. Yeah, yeah.
Right?
Mm-hmm.
But it's all contributing to that remaining kind of phase slowly.
Right.
It's very complicated.
It is, and that's the importance, I think, of modeling the dynamics accurately, because the model is kind of simulating what's happening out in the real world.
This leads to my question,
Mm-hmm.
which is actually experimentally, can you simulate this process, like creating new wastewater system in a lab? And like people that are in the 4th day of infection, people in the 5th day of infection, people in the 1st day of infection, and you contributed to the same to this system and
Right.
see how this evolves.
We've thought about that, yes.
Done or-
It has not been done, at least not that I know of, yeah. But we have, in the group that I'm in, we have kind of just had quick discussions of some kind of like experiment like that, yeah. Funding is probably the most important when it comes to that, yeah. Mm-hmm. Yep. Yeah, man. Oh, Jesse?
Oh, thank you. Incredible research, very compelling. My question is, and this is very philosophical policy, health policy, is this prediction modeling or is this modeling based on pattern? Because prediction tends to say, has this forward thing, but you're looking at data from the past and from the current.
Right.
And so I get worried when we say words like predictive modeling.
Oh,
I
I see.
Predict, and so if you could clarify or.
Yeah. So you're totally right. This is not totally prediction, as in like future forecasting, as in like retroactive kind of studying what CDC cases were looking like. Okay. Yeah, I probably shouldn't have been using the word predictive, but we do. I should say that one of the other students, Ingrid, she's working on like a forecasting prediction model with the wastewater. So I was probably using those interchangeably. Yeah. Mm-hmm. Thank you, though. Yeah.
Very small. Yeah. It's amazing. Yeah.
Matt.
Yeah, I was wondering if demographics play a role in this at all. Like, I mean, I've got to imagine there's a different shedding pattern for males and females on average. Older people.
Yeah.
Do you feel like the demographics of an area might inform the model somehow?
It's a great question. There's so many things to kind of look into. And that's, I think, the mechanistic model of the shedding curve is why it's so important to get that right. It's like, because you can change the parameters in that model to understand how that curve will change. Yeah. Okay. Alrighty. Yeah. Okay. Let's see. All right, thanks, Corey. Here you go. Thank you.
I can plug this in, or if you have a link already.
Well, come on around, take a look as long as you get this. Here they are. You want to do that? It's up to you. I'll leave it.
It's not showing anything. This is fine. need to
Do that?
use this?
Yes.
Where do I?
You can put that anywhere on your person, and I'm just going to clip this right onto your shirt.
All right, go ahead. Feels very formal. Hello.
All right, you are good.
All right. Good afternoon, everyone. So, okay. So I'm a particle physicist, associate professor here at LTU. So I'm going to talk about my research, but I wanted to make the slides available to everybody. So that's a quick link that you could type into a browser or use the QR code to get access to it. The slides have lots of different links in it, and my goal is to have these available publicly for everybody to be able to access. My work is supported by the National Science Foundation as of now. I don't know what's going to happen in the future. And I got a couple of grants. The grant numbers are over there, but so far I think it's still supported. The title of the talk is Who Ordered These? It's a little bit of a rhetorical question, and when your talk starts with a question, it's often true that the answer is either no or unknown. In this case, the answer, I have no idea who ordered these. Okay, so I'm going to talk about the physics of mesons, leptons, neutrinos. And if you don't know what these are, don't worry about it. I'm not going to try to explain the details of them to you, but I'll give you an overview. Okay. So let me just tell you the big picture of what's going on, right? So this is a picture. By the way, none of these pictures are taken by me. These are taken from open source stuff. So for instance, this I think I took from NASA. A lot of these are linked to the original sources. So what this is showing you is essentially the evolution of the universe. The universe started with a Big Bang. That's shown in that quantum fluctuations, kind of like an explosion at the beginning. It had an inflationary period, but it expanded. And then at some point, stars formed. And the way we found out about all of this is using a satellite that went out into space and measured light from everywhere, light in form of microwave radiation. And that's that WMAP satellite that you're seeing over there. Okay? So they measured everything, and then from that measurement, we kind of interpreted what's going on. And some of the things that we interpreted was that the universe is about 13.7 billion years old. That's one interpretation. The other thing that I kind of, my research kind of revolves around is the fact that we notice here there are lots and lots of stars, but there are no anti-stars. Now you might ask, what is an anti-star? So everybody knows about the hydrogen atom, right? So the hydrogen atom is made out of a proton and an electron. An anti-hydrogen atom would be something that's made out of an antiproton and a positron, just the charge opposites of the two particles. Stars essentially are glowing balls of hydrogen. They're fusing hydrogen atoms, hydrogen nuclei to form helium nuclei. And that's a nuclear reaction. Energy comes out. That's the energy that we get from even our sun. We have never seen a star that has anti-hydrogen fusing, although in our regular theory, we would expect them to form at the same rate as a regular star would. So we have never seen that. So what does that mean? It means that we're only seeing a part of the universe. We're only seeing the matter part of the universe. Now, it could be that some very distant corner there is an antimatter universe that we have never seen, but very likely is that at the beginning of the universe, there was a tiny little difference between the matter and antimatter that formed, and then eventually today, that tiny difference has shown up as a big, all matter that we see around us. So all of the things that we see in this picture, those stars, we have tried to come up with explanations of this using a particle model. And so the particle model itself is very complicated. You can see all of these, this kind of like a particle physicist's periodic table. Lots of different particles here. But what we don't understand are certain specific pictures in this universe. That's a picture of what is called the bullet cluster. And it turns out that when experimenters look at this picture, they can measure several different things. They can measure how much light is coming out, where the light centers are. They can also measure where the mass centers are. And it turns out that these are different, showing that where the light is coming from is not where the mass is. So we kind of infer from that that there must be some matter that doesn't give out light or doesn't interact well with light, and we tend to call that dark matter. If you sort of try to understand the full picture of what WMAP has measured, it turns out that this tiny segment, this big sort of several particles that that I pointed out form only a tiny sliver, the 4% sliver that's called atoms. Right? The rest of what the universe is made out of is either dark matter or dark energy, and that's like about 95% of the universe. And we don't know what that is. We don't know the nature of dark matter or dark energy. So that's sort of, these two puzzles, the puzzle of matter-antimatter asymmetry and dark matter, that's where my research, what my research focuses on. And just to give you a big picture, bigger picture of it, that's, you can look at it. This picture is so much more complicated, right? There's so many things here. And on that side, that's only 4.6% of the universe, right? So we don't know the rest of it. Okay, so I'm going to switch and talk about sort of accomplishments first, and then try to explain some of the physics. So just to give you a picture of how my work works is that I, of course, can't do experiments myself. In order to do experiments, what you would need are colliders. So these are a couple of pictures of how colliders work. So the top one over here is taken from the ATLAS experiment at CERN, where they collide protons. So the protons come and hit head-on and make new particles. And the particles are detected by all sorts of detectors. And what's going on there is sort of similar to also what's going on over here. This is, again, another detector, different picture. That's called the LHCb detector. It is a forward detector that sees the particles sort of in the forward angle. Now, so these are the schematics, proton-proton collision. Something happens, particles come out, the particles get detected. These particle collisions come from matter and antimatter matter interactions, and you can have all sorts of different things that you can talk about. Here's an example of an accomplishment. We wrote a recent paper that talks about neutrinos and non-standard interactions of neutrinos that just recently got published. I will not have enough time to talk about it. So let me just highlight some of the accomplishments of me and my students. This is one of them. Recently, this is a student, LTU student, who graduated last year, or the year before, Andrea Hauk. She was an Ed Donnelly awardee, got an NSF Graduate Research Fellowship. We wrote a paper together proposing a new method to find this matter-antimatter asymmetry through some parameters. So for instance, this parameter gamma over here, we use some math techniques, and I'm not going to go into the big words of it. But essentially what it is application of linear algebra. So classes that students take over here, they do some coding in Mathematica, and they basically, I have a code base that they add to. They create new code, and we published a paper with that. Here is a more recent paper. We identified some anomalies, and this was published in Physical Review Letters. This just November last year. And essentially what we are finding is that we're looking at specific observables, and there are a bunch of these observables, in these particle collisions, that the observables that you can measure. You find that the matter and the antimatter do not act the same way. So this is kind of going back to what could have happened at the early stages of the universe. PRL featured this in physics. They wrote a synopsis on the physics. All of these articles are linked. If you have the time, I invite you to go look at it. This is a new work that I'm working with a couple of LTU students, Luke Huddy and CJ McKenzie. CJ is an engineer. Luke is a physicist. It's a work in progress. We're essentially taking our previous paper and expanding the analysis to add additional particles to see if the same kind of inconsistency that we see in matter-antimatter show in additional systems as well. We're trying to identify possible sources of different anomalies. Okay, so let me just briefly talk about the physics. If I have a couple of minutes, I think that would work. So what's the physics? So remember I showed you that chart of the many different particles. A couple of the particles were the up and down quarks. Okay, And so these up and down quarks, they are matter particles. They interact with each other through what is called a W boson. The W boson would be an interaction particle. And this is a simple picture of that, right? Up and down interacting with the W. And the strength of this interaction is what can be measured in an experiment. And that's given by some number. And that number can, in general, be a complex number. The goal is to measure this complex number. Now, but it's not just that. Turns out that there are actually 6 different quarks, 3 up-type quarks, 3 down-type quarks. And if you take 3 things and combine with another 3 things, you get 9 things. So it forms a matrix. It forms a 3x3 matrix. And experimentally, it turns out that this one, the coupling strength over here is actually one of the largest numbers. Everything else is actually much smaller. When you experimentally try to measure a small number, it's actually difficult because you have to take more and more data to measure smaller things. So my research really goes into measuring this complex number, which is one of the smallest of this 3x3 matrix. And the phase of that, the complex phase of that number is called gamma. That's the research that Andrea did with me. There are challenges to this. So how would you measure it? Turns out these quarks, the up and down quark, they don't exist in nature just flying around. They're parts of a proton or a neutron. Or they're parts of mesons like this. This is a pi meson. That's also a pi meson. So the way you detect them is you let a pi meson decay. You find the rate at which it is decaying. And then by measuring that rate, you can kind of infer what this complex number would be. Okay, you do sort of a back process. There are complications to that process. Turns out that when you let mesons decay, they can decay to a pair of mesons. And when pairs of mesons decay, these 2 hands can interact beyond just this interaction through the single W boson. And that complicates things. So we have a way of, making that, we have a way of, a mathematical way of making that less complicated. And that's entirely what my research is about. It turns out that my research also involves penguins. This is a diagram called the penguin diagram. And why this is called the penguin diagram? It's because it looks like a penguin. If you couldn't imagine, that's the caricature of it. And the future work that I'm doing with students is to just extend this to additional particles. So this picture had a B going to a pi 0 and a k plus, but we can also extend this to a B going to an eta and an eta prime. And so that's the research that Luke and CJ are doing with me at the moment. And we are hoping to publish it within the next few months. So that's it. So questions that I'm asking, why is there more matter than antimatter? And get to talk about CP violation, but this relates to the idea that there is some CP violation. CP relates matter and antimatter. So if matter and antimatter are different, then CP is violated. That's the idea. The basic thing is that I'm creating opportunities for LTU undergraduates. They're doing research, they're publishing, they're going to graduate school, doing other things. So that's it. If you want to know more about me, I have put in a lot of the links in here. You can go there and look me up. And email me. Thank you.
Just one question. I'm not really a certain point you showed a picture. There is an image with a lot of stars. There is kind of 2 colors, blue and reddish. And then you said that from this picture,
Oh, this one.
Yes. From this picture, you said that you can calculate the light and the matter.
Yeah.
So from a picture that is an optic reproduction, that's what you see, that's it. How you can calculate the matter?
The matter. So usually what people do is that there is a technique called gravitational lensing through which they can measure how much mass there is in these centers. Not through gravitational waves, not through light. Right? So you would look at so the galaxies don't exist by themselves, right? There are things behind them. And that light curves around because of mass. And how much that light is bending tells you where the masses are. And this is just the picture, whatever that picture was, is just the picture of where the light actually comes from in that area, not from anywhere else. Does that make sense?
Yeah.
All right. Let me take my computer stuff. Oh, yeah, absolutely. I'm sorry about that. Let's do this.
Okay, thank you for sticking it out for our mini conference. I'm the last speaker. I want to talk about neuroimaging here at Lawrence Tech, in particular functional near-infrared spectroscopy. We did not have any neuroimaging capacity here until recently, although if you do cognitive science of any kind today, modern cognitive science, we use behavioral data, but frequently we include brain data. So increasingly it's important to have this capacity if you're going to be a viable cognitive science group. We were able to put together a group of people here on campus with common interests and acquire funding through the Major Research Instrumentation Program at NSF. We were able to get people from 3 different colleges. Myself and Franco and Hamad, we share a lab focusing on cognitive neuroscience topics broadly. So this equipment lives in the lab, and we're the primary users of it. There's a fairly significant learning curve involved in using it, so it makes sense for it to stay in one primary location. And then other folks, Roya Salazade from engineering, Eric Martinson had some interesting ideas about machine learning. Matt Cole and Mirat Hatat from COBIT had some interesting ideas related to consumer choice, motivation, stuff like that. So there's psychology happening all over the campus, I'm fond of saying. And you know, so people can dream up ways to look at brain data in a variety of contexts. So here's what it looks like. This is a functional near-infrared system. It's just essentially an EEG cap. It's a snug-fitting nylon cap, and it's sort of ensconced in these light sources and light detectors. So it's just in the case of this system, the light sources are LEDs, so it's extremely safe, right? You basically every other one of these is a light source. And the others are detectors of the light. The light is pulsed in to the scalp, and then it goes in and makes this characteristic shape. They always describe it as this banana-shaped path, which I think is misleading. The path is actually a sombrero. It goes in and sort of goes out in all directions, and then you can detect the amount of light that comes back. And it's depending on the distance between these, it goes just deep enough to touch and pass through the outer part of the brain, the cortex, right? So I can have a combination of one source with a bunch of detectors around there, and these are all different measurement channels, so I can understand what the brain is doing at those locations. After you collect this data and you analyze it, this gives us a pretty good idea what part of the brain was active while a person was carrying out some task, remembering, making a decision, stuff like that, right? So it's got a heat chart showing us approximately where that activity took place. Given that there are math and biology and physics professors in the room, I decided to take an extra minute to talk about what the thing actually does and how it works. The light sources, here's the electromagnetic spectrum, right? Here's visible light. This is the colors of the rainbow that we see. Here are the faster wavelengths of light, gamma rays. These turn you into the Hulk. This is radio waves are longer. The near infrared is just past red on the visible spectrum, and it's got 3 bands of its own. So near infrared, middle infrared, and far infrared. So we're working with light in the near infrared range. Some smart physicist figured out that in that range, in this range that we refer to as the optical window, somewhere between 700 and 900 nanometers, human tissue is essentially transparent to this light at those frequencies. So I can shine the laser, I can shine the LED in there. It'll pass right through the skull, it'll pass through the skin, no problem. But a lot of the light gets absorbed by hemoglobin. And the amount of light that gets absorbed by the hemoglobin changes depending on whether oxygen is bound to the hemoglobin or not. Okay? And so there's also sort of this lucky quirk of fate that here's the wavelengths, here's how much of the light is absorbed, and there's this lucky quirk that they cross in the middle here, because what that enables us to do then is we just measure light. We measure how much light's going in and how much light's coming out. That's called incident light. And then you can just solve an inverse problem to make an estimate, a unique estimate, a concentration of oxygenated and deoxygenated hemoglobin in the bloodstream. Okay? Using this thing called the modified Beer-Lambert law. What we get out of this, though, is what's called the bold signal, the blood oxygen level dependent signal. This is the same biomarker that you get out of fMRI. You're looking for blood flow in response to neural activity. fMRI, of course, costs like tens of millions of dollars. You have to have a separate building for it. It takes about $400 an hour to collect the data in there. We've got this thing we can run subjects for free all day. It's great. So the gold standard is fMRI. FNIRS is less sensitive, but it gives us essentially the same biomarker. The bold signal is a latent indicator of neural activity. What that means is we don't care at all about blood flow. We are not interested in where blood is at. What we care about is what's the brain doing when it carries out some sort of computation. So when are the neurons active? Neurons, they don't store their energy, so they essentially use oxygen to burn glucose. When they fire, they immediately deplete their oxygen supply, and then your body knows through this thing called neurovascular coupling, okay, I've got to send more oxygenated blood to that spot. And it takes about a few seconds, roughly 3, 4, 5 seconds, to replenish the oxygen in that spot. So what we measure is the bold signal, but essentially it's telling us if I see a spike in blood flow in a region, I know that a few seconds before that, those neurons in that spot were active. So the primary thing we get out of it is the spatial resolution. It tells us where the neurons were active when a person was engaged in some task. Here's a few. I'm going to go through a couple of research projects that we're using FNIRS for. I share a lab with Franco and Hamad. It's called the Computational Cognition Lab. It's just next door. My own research, a lot of it is focused on different learning systems in the brain. We have at least 2 learning systems in the brain, probably more like 3. Who knows, maybe more. And we have a prominent theory of how these learning systems work that is called COVIS, which stands for competition between verbal and implicit systems. Okay, we've got at least 2 systems that learn differently. Right, the first one is what we'll call we'll call it the verbal system. It's for explicit reasoning, things that you're consciously aware of, essentially things that you can tell somebody else. Because you're using the prefrontal cortex for that, you're using working memory. I could tell you a rule, you hold it in working memory, and you can use that to make decisions, right? So a good example is, like if you wanted to go out and to collect mushrooms, and you wanted to avoid some sort of poisonous mushroom, I could say, well, look, there's this kind of mushroom that looks just like this, but you see these little rings around there? If you see those rings, that's the deadly kind, so avoid that. So I could tell you that. I could tell you that, and you have the rule, and immediately you go off and use it right away, right? That exists somewhere in your prefrontal cortex. That's verbalizable rule. The other system involves slow associative learning in the subcortical structures called the basal ganglia. The example I like to use for this is ultrasound. Like if you've ever had the pleasure of going to see an ultrasound for a new baby, the ultrasound person goes, hey, look at this. There's the arm, and there's the leg, and there's the spine. And you're sitting there and you're going, I don't know what you're talking about. I see nothing on this. You're making this up, right? So, but that person who does that has spent thousands of hours looking at these, has become an expert at knowing what it is that they're looking at. That's a gradual learning process. It's non-verbalizable. I cannot tell you what to do. You would have to do it for a long time, and then you can, and that learning gets built up in your subcortical part of your brain. Incidentally, this is the same learning system the dog has. So when you train the dog, right, you need to have like consistent rewards over a long period of time. It has no verbal system, right? But it can learn. And that's, so we have one of those. We also just happen to have a really big prefrontal cortex, and we have this other thing. If you've ever heard of the fact, you've heard this idea that there are at least two learning systems. Maybe you heard it from this book. This was popularized maybe 10, 15 years ago by Daniel Kahneman, who's a really famous psychologist. But the thing is, not only do we have two systems, but the systems compete. So there's just two systems, but they're also both active all the time. And the example I like to use for this is, if you've ever tried to learn how to drive a stick shift, or better yet, teach somebody how to drive a stick shift, it's a nightmare, right? So I could tell you, well, you shift the clutch, you turn the thing, you do whatever. Good luck, right? It's the only way you're going to learn how to drive a stick shift is you have to get in there, and you'll kind of be right from what the person told you, but then you have to spend a lot of time learning what that feels like. You're seeing a shift from this system to this system, and those systems compete. So both systems want to learn and figure it out all the time. If the verbal system figures it out, then it just it'll dominate performance, and you'll just use the verbal system. And if it fails, despite that, you will still gradually learn. Okay, so that's how these learning systems work. What we do to probe the two systems is simple category learning tasks. We have very simple stimuli. Probably running long. We have very simple stimuli. We show them people two-dimensional stimuli like this. We say, oh look, it's this little shape, and it's got an angle and a number of bars. And I say, categorize that. Here's another one, categorize that. Here's another one, categorize that. Simple geometric stimuli, right? And you have to figure out what the rule is. And so where do these come from? These have two dimensions, an angle and a frequency. So all we do is we generate two populations of values to create these things, right? And here's one where this would be a very simple verbalizable rule. Two categories, and basically what this rule is, you can ignore the angle completely and focus, you can ignore the number of bars, you can categorize only on the basis of the angle. So like the kind of flat ones are category 2, the kind of angled ones are category 1. I could tell you that, and you'd be great at the task right away. If I take these categories and I just rotate them in space, that's the same rule, but now it's completely non-verbalizable. And you may feel like you can verbalize that rule, but I'm pretty sure you cannot. So this simple task allows me to probe how long it takes you to learn using this non-verbal system. So my prediction for FNIRS is, when I give you these two tasks, for this one, for both of them, you should have a lot of prefrontal cortex activity right away, because you're trying to figure out the rule. And in this case, you'll figure it out, and after you figure it out, we'll see sort of your activity in prefrontal cortex should plummet. But the other system, your prefrontal cortex will continue trying to figure out this rule, despite the fact that it cannot. So this will stay high. So that's a prediction that no other of the competing theories can account for. Okay, I'm probably running too long, so I'm going to try to represent my colleagues' research in 30 seconds. So Franco Delogu is really interested in using FNIRS for what's called hyperscanning. Hyperscanning is when we can actually take the system and measure the activity of more than one person's brain at the same time as they do something together. So play music or have a conversation or watch movies together, things like that. And you look for synchrony to see is it the same parts of the brain active or not. Franco's interested in competitive interactions. And in particular, there's this popular game in Italy called Mora, which is an ancient game dating back to the Roman Empire. Is this correct? This is an ancient. It's a little bit. It's like when I watch people play it, it's like a more aggressive, much more aggressive version of rock, paper, scissors. But my understanding is, from Franco, is that people can get quite good at predicting what their opponent is going to do. And our hunch is that, well, there must be some sort of a transition to this sort of implicit learning. And so that's a question that we'd like to explore with FNIRS for hyperscanning. The last little thing is, my other colleague, Hamad Al-Azhari, Hamad does research into semantic processing, right? And tries to understand how the brain processes meaning. So the knowledge we have in our brain is distributed. It's not in one spot. So concepts are kind of distributed in different spots. So you can take a concept like money, which I guess is a concrete concept. That might be one part of the brain. Something like time is more of a concept. And then a metaphor like time is money is combining those two, and people are somehow good at figuring out what this means and sort of reasoning analogically to figure out what those things mean. So we can use FNIRS to try to figure out which parts of the brain are being activated when different words are appearing in different metaphors. The last little thing, though, is Hamad also has access to a piece of equipment called a high-definition transcranial direct current stimulation system, which allows us to actually stimulate parts of the brain. So what I've been telling you so far is when you do brain imaging, usually you put people into an experiment, and you're controlling kind of what they're doing behaviorally, and then you measure their brain activity, and then you go, well, here's the part of the brain that was correlated with your decision performance or something like that. You can't really draw a strong causal inference about that part of the brain by doing it that way. It's kind of correlational. So if I have the capacity with TDCS to actually increase, what it does is it sends a electrical current, a weak electrical current into the scalp to actually increase the excitability of these neurons. It makes it easier for those to fire. I can actually change the likelihood that some region will be active or not. So I can now draw causal inferences by combining TDCS with FNIRS. Okay, I'm sorry I had to fly through that, but that was it.